Okay, so now we continue with the second part of the protein synthesis. This will be translation, in which the genetic code of the mRNA in the form of codons will be translated in order to produce the proteins. So this is a continuation from the transcription that we have discussed just now. Okay, so transcription that occurs inside the nucleus, okay, is shown over here, in which the DNA genetic information will be copied, and this will produce the mRNA. Then the mRNA will exit the nucleus through the nuclear pore. Once the mRNA is already inside the cytoplasm of the cell, then translation will occur, in which this will involve several things. Okay, besides the mRNA itself, some other things that are also important will be the amino acids. Okay, why? Because we are trying to produce the proteins, which consists of many amino acids. So the amino acid will be important, and also the tRNA. This type of RNA is important to bring the free amino acids to this site of translation. Okay, and this will be the ribosome. The ribosome will be considered as the site for the protein synthesis. Translation also includes 3 stages. Number 1, initiation. Number 2, elongation, and number 3 will be termination. Before we talk about the process of translation, we need to focus on this particular process known as the amino acid activation. Okay, in which you must understand that the amino acids are actually free in the cytoplasm. This process of amino acid activation refers to the process in which the amino acid must first attach to a particular tRNA with a particular type of anticodon. Okay, if you look in the notes, you will notice that the structure of the tRNA will be something like this. Okay, this will be the symbol for the tRNA, in which this RNA molecule will be having this special tree sequence of bases known as the anticodons. Okay, the same goes over here. You can see there are these 3 special bases, okay, at the bottom part of the tRNA, and this will be known as the anticodons. So every tRNA will be having a particular type of anticodon. Besides the anticodon, there will be another part of the tRNA that you need to know. Okay, normally it will be somewhere here. Okay, at the upper part of the tRNA, in which there will be this amino acid attachment site, in which there will be this 3' end of the tRNA that will become the attachment site for the amino acid. Okay, so this amino acid activation process will involve a particular type of enzyme known as the amino acid tRNA synthetase. Okay, so this is the diagram showing the enzyme known as the amino acid tRNA synthetase, and another thing that is also involved will be this ATP. Okay, the ATP will be important because the ATP will provide energy for the attachment of amino acid to the tRNA. Okay, and so with the help of this enzyme and also with the energy provided by the ATP, the amino acid here, the free amino acid, will be able to attach to this tRNA over here. Okay, and as a result, an activated amino acid will be formed. Okay, the activated amino acid referring to the amino acid that is already attached to the tRNA, and you should remember that the amino acid is attached to the 3' prime end of the tRNA. Okay, now we will focus on the stage number 1 of translation, which is known as the initiation. Okay, in which there will be this mRNA, okay, in the cytoplasm of the cell, okay, and comes along the small ribosomal subunit. Okay, we must know that the ribosome actually consists of 2 subunits. One will be the small subunit like this, and another one will be the large subunit. This one over here. Okay, but first, it will be the small ribosomal subunit that will bind to the mRNA within the cytoplasm. So once the small ribosomal subunit binds to the mRNA, it will start to look for the start codon, which is the AUG. Okay, the small subunit of the ribosome, okay, reaches the start codon, then comes along a special type of tRNA that we call as the initiator tRNA. So this initiator tRNA will be a special tRNA that will be having the anticodon of UAC. Okay, the anticodon, okay, of this tRNA will be UAC, which is complementary to the start codon. Okay, we know that the start codon will be AUG, and so this anticodon of UAC will be able to bind to the codon of the mRNA over here. And this initiator tRNA is also carrying a particular type of amino acid, which is the methionine. And so we can see that the initiator tRNA will base pair. Base pair means it will attach, it will combine with the start codon of AUG. Okay, after that comes along the large ribosomal subunit. Okay, and we should know, okay, that the large ribosomal subunit will be having 3 sites that is known as the A site, the P site, and the E site. Okay, so this large ribosomal subunit will align its P site to the initiator tRNA as it binds to the small ribosomal subunit. And so this will form a structure known as the initiation complex, which consists of the mRNA, the small ribosomal subunit, the initiator tRNA, together with the large ribosomal subunit. Okay, so the initiation stage is now complete. We will now continue with stage number 2, which is known as elongation. And this elongation actually requires or involves 3 repeating steps known as codon recognition, peptide bond formation, and translocation. Okay, and we will start with this one over here, the codon recognition, in which just now we know that the initiator tRNA is already attached to the start codon over here. Okay, and so what about the codon at the A site? Okay, the codon at the A site over here will be recognized by a particular type of tRNA. Let's say the codons are GGG at the A site, then a particular tRNA with the anticodon CCC will recognize that codon, and that tRNA will go to the A site and attach to the codon in the A site. That is what happening in the first step of codon recognition. Next will be this one over here, which is known as the peptide bond formation. In this particular step, we should see that the peptide bond will be formed. Peptide bond will be formed between the amino acid in the P site with the amino acid in the A site. Okay, in which this polypeptide from the tRNA in the P site will later be transferred, okay, will be moved to the amino acid on the tRNA in the A site. This step will be catalyzed by a special enzyme known as the peptidyl transferase. And we continue with the step number 3, which is known as translocation, okay, over here, in which this ribosome, okay, this whole thing will move one codon ahead, okay, in the direction of 5' to 3'. So let's say this mRNA strand over here, okay, will be having the 5' end over here and the 3' end over here. So the ribosome will move from the 5' end to the 3' end direction. Okay, the ribosome will move one codon ahead. When the ribosome moves one codon ahead, we can see that the tRNA that is originally in the P site is now transferred to the E site. Okay, can you see this one over here, this particular blue structure, okay, without any amino acids, okay. So this tRNA, okay, which is initially in the P site will be transferred to the E site, and once the tRNA is in the E site, it will be released from the ribosome. Meanwhile, the tRNA with the polypeptide in the A site, which is this one over here, will be transferred to the P site now when the ribosome moves one codon ahead. And so the 3 steps of the elongation, the codon recognition, the peptide bond formation, and the translocation will continue, okay, in a cycle in which after translocation just now, okay, after translocation, the steps of codon recognition will occur again, and this will be followed by the peptide bond formation again, and then it will be continued with the translocation again, and these steps will be repeated, okay, until the polypeptide chain is completely formed. The elongation stage will eventually stop, okay, with the stage known as termination. Okay, when does this happen? This will happen when the ribosome reaches a stop codon. So along this mRNA, there will be one particular stop codon. Okay, any one of the 3 that we should know now. Okay, what will be the stop codons? It might be the UAA, it might be the UAG, it might be the UGA. Any of those 3 stop codons. Okay, so if the ribosome reaches the stop codons, there will be no tRNA that will recognize the codon at the A site, because there will be no tRNA that will be recognizing the stop codons. Instead, there will be a protein known as a release factor that will bind to the codon at the A site. Once the release factor binds to the stop codon, okay, the polypeptide chain will be released from the ribosome. The tRNA will also be released from the ribosome, and finally the ribosomal subunits, everything will dissociate. The important thing now is the polypeptide chain is already produced. This is the proteins, and this is the protein molecule that is synthesized through this process of translation.